series of Stanford University, a constellation of sort of superstars. Today we have Anshul Kundaje and students, Abhimanyu Banerjee and Daniel Kim. And tomorrow, just a few more announcements. So tomorrow tomorrow at 2 p.m. we have Will Greenleaf in his lab presenting. And then we go on March 15th and March 22nd with the regular seminar series. As with all this, this is a closed meeting, really. So we'll be hearing unpublished results. And like with our local colleagues, please follow up with Anshul and his colleagues. If you have anything you want to follow up on, please talk to them directly. So a few words on Anshul. Anshul Kundaje is an assistant professor of genetics and computer science at Stanford. He completed his PhD in computer science from Columbia, and then he postdoced at Stanford, and then continued along the same lines as a research scientist at MIT and also at the Broad. So he's at the Broad for between 2012 and 2014 before starting his faculty job out in Stanford. Anshul's recipient of multiple awards, including the 2019 Chen Award for Excellence in Human Genome Organization from the Human Genome Organization, and an NIH Director's New Innovator Award and a Sloan Foundation Fellowship. So Anshul's lab develops statistical and machine learning methods to integrate non-coding regulation and variation. And I'm personally very grateful to Anshul. I don't know if I've ever thanked you for this, Anshul, but our team here, Chuck Epstein and Noam Shores, the team generates an enormous amount of data for ENCODE and for the roadmap. And Anshul has been the most generous colleague in these consortia, going all the way from making sure that, sanctifying and, quality controlling the data in incredible detail, but then going all the way through to the really sophisticated integrative analysis and machine learning methods that have really, I think, amplified the impact and accessibility and importance of all the work and all the data generated in these consortia. So I'm very grateful for Anshul and delighted to have you here presenting to us. And I'm sure we'll learn a lot more about the latest in genome characterization by these computational methods that you're bringing forward. Thank you for presenting, Anshul. I'll let you give your start and then turn it over to your team.
Great. Thanks, Brad, for the nice introduction, and everybody for the kind invitation. Great to be virtually back at the Broad. Hopefully, we can do this again in person sometime in the next year. Today, let me just share my screen. Give me a second. All right. Hopefully, you can see everything.
All good. Yep.
Okay. Right. So I'm going to start off with about 30-minute session, and then I'll have two of my students present some new work. The part I'm going to speak about today is sort of a new framework that we've developed. This just recently got published 2 weeks ago in Nature Genetics. It's sort of as you know, my lab has been, working on new approaches for understanding genome regulation, computational approaches, and over the last 4 or 5 years, we've been pretty fascinated by these deep learning models, and probably for the wrong reasons, in the sense that deep learning typically has been characterized as these extremely powerful black box predictors, where the primary use of them is to make extremely accurate predictions. We sort of got interested in these models because of the ability to learn, de novo representations. That is, you can often feed them very raw forms of data, inputs and outputs, and they learn mappings, predictive mappings from various kinds of complex inputs like sequences and images to various kinds of complex or simple outputs like different kinds of labels. And the cool thing is not just that they can make accurate predictions, but that they can do so by learning powerful representations de novo. And so the idea was that if these models actually work for genomics, mapping uh, DNA sequences, regulatory DNA sequences to various kinds of readouts, maybe if they're able to do this very accurately, maybe they actually learn representations that can reveal novel biology. And what we are really interested in is a problem that I think is still unsolved and is a very hard problem of trying to understand the syntax of regulatory DNA. Okay, so I will talk about this today, and then we will see some follow-ups on this work from my students, where they will take these or similar frameworks and use them in many other interesting biological settings. So this particular work was a fantastic collaboration with two people who are not in my lab. Ziga Avcek, who actually was a summer student who visited us a few years ago from Technical University of Munich, and this idea just started off as sort of a fun exercise. Let's see if this works, and it just kind of went off the rails and became something way more amazing than I think any of us in this team would have imagined. However, what I'm going to present today is built on years of work from Avanti Shrikumar, who was a graduate student in my lab. She graduated just a few months ago, last year. She has built a whole suite of methods for interpreting neural networks, which we will repeatedly look at today. Um, Melanie Amar also contributed a lot to this effort, but I also really want to thank Julia, because there's no way we could have done what I present today without an expert biologist in genome regulation working completely hand in hand, like 50-50, extremely invested and really passionate about the methods, just as we are about the biology. So to me, this is like my favorite project of all time. And it's just like very satisfying to see a computational group and a like a solid biology group collaborating and taking the whole product to essentially a new level. And of course, I should thank my funding sources and a lot of other people who helped with data generation and various kinds of follow-up validation experiments and so forth. So just getting back to the crux of the problem, as you know, we can generate a whole variety of different kinds of experiments for profiling regulatory DNA, such as ChIP-seq experiments for transcription factor binding, DNase and attack-seq experiments for chromatin accessibility, and of course other kinds of experiments to profile chromatin as well, such as ChIP-seq and cut and run and so forth for histone marks and other kinds of readouts. This is a snapshot of some ENCODE data um, from Bob Thurman's 2012 paper. Um, Given this kind of data, I think it gives us a real opportunity to not only map regulatory elements, for which there has been a lot of work already, but to really understand how the DNA encodes all of this interesting diversity of molecular activity. And the question as I said, we're really interested in is going beyond just motifs. So as you know, transcription factors bind these regulatory DNA sequences, and most TFs have specific sequence affinities, so they recognize specific motifs. But in vivo, these factors bind DNA not independently, but often in extremely cooperative and competitive ways, including competition with nucleosomes. And so we really wanted to understand the higher order organization of regulatory sequence that is trying to understand what we refer to as motif syntax, rules of arrangement, combinations that appear together, preferred spacing, orientation, and not just like the rules, but to really understand how these rules might affect cooperative binding. So we want to discover these rules and their effects directly on TF occupancy. So to do this, we actually model the problem as a machine learning task where if you're given a profile across a genome of some experiment, let's say a TF ChIP-seq experiment, what you end up getting is, you can think of taking the genome and binning it into chunks, let's say of 1,000, 5,000, 10,000 base pairs. Each of those little sequences is affiliated with a signal, right, at each base pair in the genome. So what you end up with is millions of chunks of sequence associated with corresponding labels coming from the experiment. And the classical approach to model this kind of data has been to take these chunks of sequence and map them to scalars. So you take the signal over this sequence and you either map it to a binary label. For example, you do some kind of peak calling on the data and you say, there's a peak or no peak, or it's active or inactive, or bound or unbound. And so you typically summarize the whole sequence by a scalar label. Or you summarize it with some real value number, which corresponds to the enrichment or some such number. And so you solve this as a classic classification or regression task where your goal is given a bunch of sequences, you want to map them to labels. And there's been a ton of work in this space for decades or more, um, including biophysical models, machine learning methods like support vector machines, and even neural networks since 2015. Um, we decided to take a slightly different approach. These profiles that you get from the data at base resolution, like if you look at the read counts, the 5' ends of these reads at each position in the genome, you get very sparse signals, right? So typically you have to aggregate them in windows to get enough power to call peaks and things like that. But we just realized that, I mean, if you really look at the base resolution, profiles of these data, ChIP-seq, ChIP-exo, DNA-seq, ATAC-seq, um, you will often see really exquisite structure, right? So when the protein binds the genome, the fragments that are sequenced by the ChIP-seq experiment or using exonuclease digestion, you end up getting these really beautiful, symmetric or asymmetric mirrored footprints or profiles on the two strands for ChIP-seq and ChIP-exo. For DNAs and ATAC-seq, if you just look at the strand agnostic coverage profiles of the 5' ends, you end up with, again, beautiful profiles that often capture very specific footprints of these transcription factors. So we thought, like, let's see if we can, this was the crazy idea which we had, when Ziga visited us many years ago. Why not just like avoid processing the data altogether and just model it at its raw resolution? Like, let's just like take a sequence right here. Let's just take the DNA sequences, and rather than trying to take the signal over the entire 1,000 base pair bin or whatever it is and summarize it into some scalar through some peak caller, let's just like take the sequences and predict the base resolution counts of 5' ends of reads on the plus and minus strand. Let's just see if that works. There are obviously like serious challenges to this, which is why we are, not convinced this would actually work. And the biggest challenge actually is the sparsity of the data. If you ever take, any of these data sets, ChIP-seq or whatever, and visualize the 5' ends of reads at base resolution, they can often look like extremely, noisy and sparse because we all don't sequence these data sets extremely deep, and there's a limited library complexity. But we thought about this a bit, and the way we try to get around it is by training this neural network, which is, the architecture is not at all complex. It is very standard. It's just a convolutional neural network with multiple layers. We have some fanciness in there. There's these things called dilated convolutions, which allow you to learn deep models with minimal parameters. And we have these things called residual connections, which again allow us to learn deeper models um, more efficiently. But it's a very simple architecture. Otherwise, these convolutional neurons in the neural network, basically they are pattern detectors. They are motif-like pattern detectors. The first layer learns simple patterns. The next layers combine those patterns to higher order patterns, and you can keep going, right? And so in this particular case, at least for ChIP-seq and ChIP-exo, we started off with 1,000 base pair sequences or 2,000 base pair sequences and decided to map them to 2,000 base pair profiles. So think of it like a text to speech converter sort of thing, right? Like the text is the sequence, the speech is the profile that the experiment is generating. And there are some additional, like the really clever thing that Ziga did was, as I said, accounting for the noise in the data. It's super sparse. And so if you apply typical, error functions that people use to train these models, they would very unlikely work because the zeros that you see in these profiles are not real. I mean, they're not real zeros. They're just artifacts of undersampling. And so we developed sort of a loss function called the multinomial negative log likelihood, which really accounts for this process of a bunch of reads falling onto the, let's say there are n reads falling on 1,000 base pairs. And typically you just quantify n. You map the sequence to n reads. That's usually the scalar output that you're modeling. We say, okay, we'll not only try to predict the n reads that fall onto the sequence, we will also try to predict how those n reads are distributed across each of the 1,000 positions. And that distribution is like, like a balls in bins problem, right? You have 1,000 bins and you've got a bunch of n balls and they're distributed across these bins. And if you think about balls and bins, the first thing that comes to your mind is the multinomial distribution, right? So the multinomial distribution captures that kind of noise really nicely, positional or binning distribution of a bunch of n items. And that's really what we use to model the error in the signal. And that makes a huge difference, and you'll see in a few minutes why. So to test out this model, we wanted to take a system that really kind of allowed us to push the models to its limit. And we decided to pick a very well-studied system, 4 classic pluripotency transcription factors, OCT4, SOX2, NanoGen, KLF4 in mouse embryonic stem cells. And Julia's lab actually developed this chip nexus assay, which is sort of a development on chip exo, making it more efficient and higher complexity. And she generated this beautiful chip exo data, chip nexus data in mouse ESLs. And we decided to go with chip nexus because we really wanted to see, like, the footprints that you get from chip exo. We wanted to see if we can really leverage that and do, substantially better than we could have with lower resolution assays like ChIP-seq. But as I'll show you in the next, towards the end of the talk, and Abhi will show you this in his talk, this method works really well on all kinds of data, not just chip exo, but on ChIP-seq, on DNAs, attack, histone ChIP-seq. We've applied it on ProSeq and GrowSeq and Rampage and Cage. Any regulatory profile, this model seems to work really nicely. So we trained our models on this data. And the way we evaluated the model is through a simple cross-validation setting where we held out entire chromosomes. We train the model on a subset of chromosomes, and we predict on the entire remaining chromosomes. And we do this in sort of a 5-fold cross-validation setting. When we do that, these are some examples of what the predictions look like. Kind of slightly mind-blowing when we first saw these results, like in 2 weeks after Ziga came to the lab. Again, he's an incredible human being. Just cannot believe this project actually worked out. But every time I see this figure, it just kind of blows my mind because I remember seeing something very similar to it, like 3 weeks into his summer rotation, summer visit. But this is what it looks like. I mean, these are 3 enhancers in the genome. This is OCT4, SOX2, NanoGen, KLF. These are the observed signals from the actual chip exo data, and below it are the predicted signals from the model on held out chromosomes, basically never seen before. So you can see in many cases they are indistinguishable. They look really similar, including the exact positions of these footprints. In other cases, they differ a little bit, and there are other cases where the model actually denoises or desparsifies the data, as you can see right here. This is an example of an enhancer where the coverage is slightly lower. You can see a bunch of these zeros popping up, fake zeros. And the model, is able to impute because it's predicting the expected value of this multinomial distribution, and thereby sort of predicting missing information. And it becomes even more obvious. I'll show you towards the end of the talk. For ChIP-seq, the denoising is kind of spectacular. So if we just, these are of course some 3 examples.
I'm sure, just to be so I'm totally clear, the model is only using DNA sequence of the loci?
Correct.
And you're having it look across the entire genome?
Correct.
Or I guess you first train it on some chromosomes and then you
Correct.
have it.
Yeah. So I should clarify here that the goal of this effort is not to predict binding in some new cell type, new condition. That's not the goal here. Those kinds of models also exist and they're great. Our goal here is to fit an extremely accurate model to an existing assay you have, such that if I start manipulating the sequence in that same cell context, I can make very accurate predictions. Okay. And a way to test that is to just hold out entire chromosomes where sequences that you've not seen before, and then you make predictions. So that's really the cross-validation setting right here. And the predictions are not going to be important beyond the next 2 slides. Okay. We're going to focus.
I'm going to say people can put questions in the Q&A function on here, and we'll read them out.
Yes. So So these are these sort of three examples, but we wanted to really evaluate the performance of the model, genome wide. And so, you know, we looked at two performance measures. Don't worry about exactly what they mean. There's a Jensen-Shannon divergence and the multinomial log likelihood. Both of them measure the how similar the predicted profiles at base resolution are to the observed profiles across across peak regions across the genome. Um, and on the x-axis, you're seeing sort of the profiles being evaluated at different resolutions, at single base pair resolution, smoothing with 2 base pairs, 5 base pairs, 10 base pairs. So the smoothing allows the profiles to be, not exactly the same at base resolution. Due to sparsity effects, we smooth a little bit to see how performance changes. And the lower the better. Okay, so for both of these performance metrics, the lower the value, the better the predictions. So the red curve is actually our BPNet model, and we wanted to compare our predictions to a performance to some upper bound and lower bound. So we looked at two bounds. The best you could potentially do, well, one upper bound is if you did very high quality replicate experiments, right? You did the same experiment twice and you measured how similar the replicates are to each other. That's the blue curve. Okay. So what you can see is that the red curve is often very close to the blue curve, right? So we're really close to replicate level concordance, and we are substantially like better than other baselines, like let's say a randomized predictor where you just randomize the profiles and you compare the randomized profiles to the true profiles. That's the worst predictor you could get. And you can also just compute like the average profile across all the peaks and use that as a predictor for all the peaks, and that's the green curve. Okay, so it's substantially better than the baselines, and we are really close to replicate concordance. um, so that's we are pretty happy about that. So okay, now we're happy that our models can make accurate predictions on unseen sequences. How are they doing it? That's the big question. So as I said, Avanti has built a whole suite of methods over many years to address these kinds of questions for neural networks in general. So what we do is we take this deep lift method that she developed in 2017 and we adapt it for these profile-like outputs. And basically what it does, deep lift does, is takes the output of the model, some sequences given, the model makes a prediction of the profile. It takes an output and it recursively decomposes the contributions of these individual pattern detectors all the way back down to sequence. So you end up with base resolution contribution scores of every nucleotide in the genome to the corresponding profile the model has predicted. And so when we do that, this is an example again of the OCT4 distal enhancer. You can see that there are all 4 transcription factors bind the sequence. Some of them have nice sharp footprints, others have more diffuse footprints. And if you take the model and you infer these deep lift scores, you get these really beautiful base resolution contribution scores, and it really highlights classical things like the OX-SOX motifs and SOX2 and ZIG3 and NANOG and KLF4. So the same enhancer is being quite obviously differentially recognized by the 4 different TFs. These 4 TFs are all jointly binding the same enhancer, and they're using different parts of the sequence. So the nice thing about this approach is you can take a piece of the genome and you can interpret it in the context of whatever profiles you're interested in. And this is quite nice because you can not only do it for these 4 transcription factors in one cell type, you could also take one transcription factor and look at how its recognition code changes across cell types, right? Or you could take DNA-seq, attack-seq, and chip-seq and compare how all of them are translated from the same sequence. So this is quite nice because you can now make these very kind of high-resolution predictions of individual enhancers in the genome. But then we wanted to kind of summarize this across the whole genome. And to do this, we Avanti developed this TF Modisco approach, which computes these deep lift profiles for every binding region, every peak for each of the transcription factors across the entire genome. You get these contribution scores of every nucleotide to the predicted profiles. You throw away, the pieces of the sequence that are not predictive, and then Modisco basically runs a clustering algorithm that takes these seclets, these little sequences, aligns them, and collapses them into non-redundant motif representations. And we learned 2 types of representations, one of which, for the motifs, one of which is called the contribution weight matrix, CWM. And what this does is actually summarizes the average contribution score of every base at every position in the motif, okay? Which is different from the position frequency matrix or the position weight matrix, which we are quite familiar with, which summarizes the frequencies of nucleotides, of bases at each position in the sequence. And this will become very important, not only in my talk, but also in Abhi's talk. Because what we'll show is that frequency is not the same as predictive importance, okay? And that's a pretty important contribution here from us. So if we do this for all the 4 transcription factors, we end up with actually a pretty large slew of motifs. It's not just 4 motifs for 4 TFs. We actually need to get about 50 motifs to explain binding for 4 transcription factors. And in many cases, we pick up, I'm just showing you a few of them. We pick up classic, the heterodimers, monomers, homodimers, um, multiple versions for KLF4. We pick up multiple versions for the nanog motif, which I'll get to in a second. And we also pick up indirect tethering events with ZIC3, with really interesting like TF3 box. And we have cool vignettes in the paper as to what these might be doing, including validation for some of them like ZIC3. So there's indirect recruitment to motifs of transcription factors that we not even assayed, ESRRB as well. And the important thing is we can take these motifs and really dissect their contribution to binding of each of these 4 transcription factors. So, motifs like OX2, the heterodimer, have significant influence on almost all 4 transcription factors with different footprints. Some of them are very specific, like the nanog motif is very specific to nanog, the KLF4 motif is very specific to KLF, but you see this very interesting kind of um, contributions or motifs to all 4 transcription factors in different degrees. And so this is sort of the first evidence of kind of interesting cooperative effects where one motif or one TF influences binding of another TF. We are able to map, the precise motif instances, and that gives us a nice way to really at high resolution annotate these regulatory elements. So I'll just jump to the nanog motif. And as I mentioned, we actually identify 3 different versions of the nanog motif. And this will be a theme that Abhi will explore further in his talk. Um, and in this case, then the 3 motifs, they share this TCA core, which has been shown before to be, which is what we call the main footprint. But then we see the flanks actually showing different degrees of sequences corresponding to these 3 different binding modes. And what's very interesting is that each of these pieces actually has been previously validated in different settings. So there are like EMSA assays that are validated, the AATGGC portion. There's a crystal structure that is validated this sequence as well, the GGAATGGC portion for nanog. That's a crystal structure for monomer of nanog binding. And so our initial hypothesis was that these longer versions of the motif are likely capturing homodimers. Um, they're capturing multiple narrow units, binding the DNA in very specific ways. So there was some support from previous experiments and like the crystal structure as well as EMSA and CLEX that each of these portions of these motifs are likely valid. So we learned some novel motifs, but as I said, our primary interest was to dissect syntax. And so we started off by looking for what often people refer to as strict syntax. By strict syntax, we mean like very precise spacing between pairs of motifs. Okay, and on the x-axis here, you're seeing the frequency of pairs of each point here is a pair of motifs. And on the x-axis, you're seeing um, the top spacings by various spacings, and y-axis is the frequency of those spacings for each pair. And so things towards the left are pairs of TFs that um, show extremely strict spacing constraints with high frequency in the genome. And so there are a few, as you can see, that show strict spacing. Most of them happen to be, as you can see right here, most of them happen to be classical physical composite motifs of homodimers or heterodimers, like nanog with itself, OCT4 SOX2 nanog with like 8 base pair spacings. These are well known, like protein-protein interactions that directly bind DNA. But we also saw these other weird like combinations that were essentially like really distant apart, like 46, 68, 71 base pairs with strict spacing. But when we looked at where these kind of instances lie in the genome, they all exactly overlap repeat elements. Okay, so they're all coming from retrotransposons. We see no other instances of strict spacing besides the really short range ones, other than these pairs coming from a retrotransposon. So these TFs, as you know, bind these transposon elements, which are actually found to be enhancers in many cases. But the really cool thing is that our model is able to distinguish the precise positions of the motifs contributing to binding, even within these repetitive elements. So this is really the difference between a position frequency matrix and a contribution weight matrix, where the PFM, if you just look at the PFM, you actually, I mean, this is an exact, almost exact repeat that is present in multiple thousands of regions in the genome. And so if you try to derive a PFM, you just end up getting the entire repeat basically lighting up. But if you look at the contribution scores, the contribution weight matrix, the neural network precisely tells you like which parts of that repeat are being utilized by the different TFs. And Abhi will also really leverage this property to dissect binding affinity of another class of transcription factors, the C2H2 zinc fingers. So that's really cool because now we can take all of these retrotransposon families and we can start dissecting individual instances for the precise motifs driving binding. The other cool thing we found is if we take the nanog motif, the core nanog motif with the TCA core, and we look at flanking sequences. If you look at the PFM, you don't see much. You just see the TCA core motif popping up. But if you look at the contribution weight matrix, which summarizes the importance scores, you see something really interesting. You see these plumes of importance pop up every at a regular frequency, these TAAT patterns. And the really cool thing is if you look at the frequency of these plumes, it happens to be exactly 10.5 base pairs, which is the helical turn of DNA. And what's interesting is there is evidence that homeobox proteins like nanog can in fact bind even nucleosomal DNA as homodimers in NCAP CLEX, even in vitro data. And what's really interesting is these kinds of frequencies of flanking nucleotides we see as a specific property of nanog, not just not just for all TFs, but it's very specific to nanog. So all the motifs of nanog, all motifs that contribute to binding of nanog, as learned by the model, end up having this really interesting 10.5 base pair frequency. This is the power spectrum of the 10.5 base pair, or sort of the this sort of important signal, and you can see like an amplification of the frequency of the spectrum at 10.5 base pairs specifically for motifs that drive nanog. So if the SOX2 motif is helping drive nanog, it has a 10.5 base pair frequency next to it. But if SOX2 is primarily driving SOX2, it doesn't. So it's a very context specific importance or frequency effect that seems to be recognized by nanog. So we decided to look into this a little more. And if we take nanog motifs, just the cores, and we look for positional spacing between these, we don't find hard syntax. That's the reason in the strict spacing analysis, we never really saw nanog with itself at some very strict spacing constraint. But what we do see is that if you just plot as a function of distance, the frequency of nanog pairs of nanog motifs, you again see this really beautiful 10.5 base pair positioning frequency, but it's a soft syntax. There's preferential positioning of these TCA sequences at multiples of 10.5 base pair. And that's independent of the orientation of the motifs. It could be on the positive strands or negative strand, positive, negative, it's the same. And the nice thing is if we take the data and now we align them with the motifs, right, with these sequences that have these 10.5 base pair patterns, actually you see that this signal is in the data. This is the chipped exo reads. You can really see once you align these sequences with each other, you can really see that there is a 10.5 base pair, spiking of the signal. And that's really what the model is picking up. Okay, and that's how it's able to learn this soft syntax, because it sees these spikes in the data, it sees these spikes aligned with the TCA sequences, and it learns that relationship de novo, and we're able to extract it from the data using our interpretation methods. As I mentioned before, this sort of this soft syntax, soft helical syntax, is not just for nanog with itself, but nanog with all the other motifs, with SOX2, with OXOX, with nanog, with differing degrees of strength. But across the board, you again see nanog having this 10.5 base pair preferred soft syntax with itself and with a whole bunch of other TFs. So one important question is, given this syntax, can we infer actual causal cooperativity from the model, right? Because the syntax doesn't tell us if it's doing something. I mean, it's in the data. That's what the model's telling us. It's in the sequence. But can it really, how can we dissect its influence on cooperative binding? Typically, the way you do this is through an actual experiment where you perturb sequences or generate synthetic sequences with different configurations of motifs, and then you do experiments to see if you remove one motif or you add one motif or you change the spacing, how does it affect binding of one TF versus the other? And so that's actually exactly what we did, except we did this um, in silico. So we start off with synthetic sequences where we inject motifs of two kinds or of the same kind, and then we can change spacing, and we can use a model as an in silico oracle. Since it's so predictive, we can use it to predict what the binding profiles would look like. We can also do this in the genome itself. So we can take genomic enhancers and we can annotate all the predictive motifs that the model finds, and we can start systematically deleting them like a CRISPR experiment, and the model would predict what would happen to binding. So we did this, and here's one example. This is OCT4. This is a synthetic sequence where we embedded the nanog motif at this position and the OXOX motif at this position. And now we start moving the OXOX motif towards nanog to try to understand the influence of spacing on these two TFs. And as the motifs are moving towards each other as a function of distance, we are plotting or measuring the change in the binding occupancy as measured by the predicted footprints from the model for both TFs. Okay, so the red curve is the change in the binding occupancy for OCT4, and the golden curve is the change in binding occupancy for nanog. And you see something very spectacular pop up, which is that the model predicts that as a function of distance, the binding of OCT4 does not respond at all to the nanog motif, except when it comes really close by. Okay. Whereas on the other hand, if you look at nanog has a massive amplification, almost an exponential rise, as the OXOX motif comes closer to it, all the way starting from 150 base pairs, which is the sort of the approximate size of a nucleosomal turn of DNA, and it gets rapidly amplified. And as you get closer, you see this again, this 10.5 base pair frequency really lighting up. So what the model is saying is that there's a massive cooperative influence of OCT4 on nanog, all the way up to 150 base pairs, which gets amplified exponentially. And as you get closer, you start getting a very nice preferred syntax effect. That is, at multiples of 10.5 base pairs, when they're on the same side of DNA, they really have a strong cooperative effect. But we can replicate the same, as I said, the same analysis on the genomes, enhancers in the genome, where we take enhancers and we mutate motifs. So here, for example, this is the OCT4 enhancer again. We have the OXOX motif and the nanog motif. We can mutate the OXOX motif, and the model predicts that OCT4 binding dies and nanog binding dies. And we can mutate the nanog motif. You can see the OCT4 binding stays pretty much intact. And nanog binding actually gets attenuated, but doesn't get destroyed completely. The OXOX motif can, in fact, indirectly recruit nanog. So this, again, is asymmetric relationship, OCT4 acting as like a classic pioneer factor, kind of binding independently, and nanog really depending on OCT4 for its binding. And if we plot this across all the enhancers in the genome, we again pick up the same asymmetric cooperative effect with this 10.5 base pair preferred spacing. So we learn a really interesting, these kinds of cooperative effects between all pairs of motifs. And you can see that some of them are pretty short range, what we refer to as direct interactions. Others are much more long range, as you can see right here. They go up to a nucleosome range. So we can learn these syntax-dependent cooperative effects, pairs of TFs, and causal effects of one TF on the other dependent on spacing, orientation, and even affinity, which we started doing more recently. I'm going to stop in 2 seconds just to hand it off to my students, but just to show you, these are all predictions from the model. So we actually did some CRISPR experiments to validate the syntax. One of the first things we did is we took, especially nanog and SOX2, which showed also this kind of really interesting cooperative influence with respect to each other. Here's an enhancer where you have SOX2 and nanog binding. This is the motif that the model predicts as being important. Um, if you take the observed and the predicted, they look really similar at base resolution. We then mutate the motif at these two positions that the model predicts are really important. Um, and the model predicts that the footprint gets attenuated not just at this position, but also at this position, which is quite a distance away. We perform the experiment. That's kind of precisely what we see. We see attenuation at this position and attenuation at this position. So this kind of validates our kind of medium range cooperative influence of SOX2 on itself. We can then test the effect on nanog. So the same mutation, how does it affect nanog? And once again, you see that the predictions from the model really quite nicely recapitulate what you see in the experiment. You see kind of a global attenuation of nanog binding across the entire enhancer by deleting two nucleotides in the SOX2 motif. We can then do this for the nanog motif, which is sitting next door right here. And we're looking at now the mutation of the nanog motif or nanog binding. You see attenuation locally, which is very short range, right? So you see up to this range. And then if you actually look at the effect of the same motif mutation on SOX2 binding, the model predicts no effect. And the experiment also shows basically no effect. Just right here proximally, there's a slight decrease in the footprint, as you see here. But globally, SOX2 doesn't care about nanog. So we've also done another bunch of, I need to hand it off, so I'm not going to get into this, but we've validated sort of that these models also work. The binding models can also generalize to predictions of differential changes in chromatin accessibility genome-wide after depletion of OCT4 and SOX2. And we've also checked whether the model can predict effects of sequences in report assays, and actually it does really well in those scenarios as well. So the binding model actually generalizes the syntax that the model has learned for binding appears to be functional in quotes for other readouts as well, such as chromatin accessibility after transdepletion of these TFs, and also for reporter experiments. Okay. So just to summarize my talk here, I showed you a BPNet, which is a new model that can map DNA sequences to base resolution profiles. We can actually do this for a whole slew of kinds of data, which we started doing now in the context of ENCODE. These are not black box models. That's the reason I showed you this little cartoon right here. You can open up the black box and really discover very interesting biology, soft syntax mediated directional causal TF cooperativity, which is really cool. Never really imagined we could do this with these kinds of models. And I showed you that the syntax of TF binding is predictive of CRISPR mutation experiments, where we actually perturb motifs and look at effects on binding. The syntax is predictive of differential chromatin accessibility after TF knockdown. And the syntax is also predictive of reporter expression activity of enhancer sequences. So I'll again just thank everyone who's involved in this study, and I have a few minutes for a few questions.
Anshul, this is amazing. Thank you so much. I really enjoyed that. Can you just remind me how much time we want for Abhimanyu and Daniel's talks? Because we have so many questions. I know we're not going to get through them all.
Yeah, it might be, maybe we just take 2 questions. I think each of them has planned about 20 minutes.
Okay, let's do that. And then what I will suggest is that you can actually type your answers in the Q&A, and if there's anything, we can return to at the end if we have more time. So I'll just jump in with a couple of these then. So Adam Rubin asks, how do you interpret the cases where BPNet outperforms the replicate comparison? And does that suggest that the model is overfitting to experimental replicate specific features?
Great. That's a very good question. So the question of overfitting is not, just remember that everything I showed you, the performance is all on unseen sequences. So the question of overfitting would happen if the model's performance on the training set was much higher than the model performance on the test set. And that's not what we see. We actually see that they are very comparable. The reason you see that effect with sometimes the model beating the replicates is because the replicates are actually sequenced at half the depth of the data we actually trained it on. Okay, so on the training set, we actually pool the replicates and we train the model on the pooled data. And so technically, the ground truth that we see, the model sees, is actually twice the depth of the individual replicates. When we're comparing on the test set, we are also comparing the model to the deeper sequence data, right? That's our that's our model's performance, but the replicate concordance, unfortunately, happens to be at half the depth because that's that's the depth we looked at. If you subsample the replicates and you compare them, then they basically line up really nicely. So it's basically an artifact of depth. So that's why I don't like to use the word that our model has superhuman performance or whatever it is. It's actually as close to replicates as it gets. So the overfitting problem would have been surely problematic if on the training set we got like perfect fits, on the test set we got kind of crappy fits. That's not what we see. If you hold out entire chromosomes, the training and test performance is very much the same. So it's not really overfitting, it's actually able to pick up those footprints quite nicely.
Thanks, Anshul. So I'm gonna try and pick off one topic that a lot of people have asked about, and that's the data you need to train the model. So Jason Bornrosser says this work is amazing. This is going to change how we think about TF binding, but one challenge is that ChIP-Nexus is really hard. And do you know how much you could learn relative to ChIP-Nexus for other high-res approaches like cut and run or cut and tag? And other people are asking similar things like single cell attack. How much data do you need to train the model? So I wonder if you could just comment on the data needed.
This is just a snapshot of what the model does on ChIP-seq. Okay, so this is these are two different binding sites for E2F6. This is ChIP-seq. You can see how crappy the data is. I mean, I don't mean this in a bad way. It just really is very sparse. It's amazing data. Don't get me wrong. But this is what the model predicts. Okay, this is the model's denoising of ChIP-seq data. And these are two different ChIP-seq data sets with two different antibodies. This data set has half the number of peaks as this data set. If you look at the observed tracks, they look completely different, meaning at base resolution. But check out the model's predictions. They are literally like replicates. So we've trained this now on hundreds of ChIP-seq data sets, from data sets having like 300 peaks. I mean, Abhi will show you in his talk, like we've trained this on really sparse data sets, and the models still learn really well. So you can apply this to, we've applied this to ChIP-seq, cut and run, DNA's attack, DNA's histone ChIP-seq. It works across the board. So it actually works out very nice. It trains in like 2 hours, and on a single GPU on your laptop. It's not really memory hoggy or anything like that. It just seems to work out of the box.
Amazing, thank you. So Anshul, what I'm going to ask, if it's okay, is while we're going through the other talks, there are a lot of really good questions, and you can type in the Q&A and answer them, and then everyone will be able to see it. So if there's any you want to answer, typing your answers, you can do that. People who put your questions in the chat, if you could paste them into the Q&A, and then Anshul will be able to see them and answer them in the Q&A if he has the time. And if not, we'll try and return to them at the end, but I'm not sure we're going to have a ton of time if we've got 2 more talks to get through. But thank you so much, and thanks to everyone who posted questions. All right, Anshul, I hand it over to you to introduce your folks.
Great. So I'm going to introduce one of my students in my lab, Abhimanyu Banerjee. So he's actually in the Department of Physics. He joined a lab a few years ago. He's hopefully going to graduate this year. So he's definitely potentially interested in postdoc opportunities as well. So if you're interested, definitely reach out to him. He's going to present on how we've used these models to dissect really interesting alternative binding modes of one of the largest classes of transcription factors in the human genome, the C2H2 Zinc Fingers. So Abhi is going to present that, and Daniel's going to present on how we've used similar models to dissect in a dynamic system using attack-seq data, chromatin dynamics and skin differentiation. So take it away, Abhi.
Okay, thanks Anshul for the introduction. I'll just share my screen so you can see it, right? Yeah. Okay, so thanks Anshul for the introduction. So I'll get started. So today I'm going to talk about alternative binding motifs for C2H2 zinc finger transcription factors. So right, so the C2H2 family of zinc finger TFs is the largest family of TFs in humans. It's basically half the human TFs. There's around 800 such TFs in 800 such TFs basically. And the thing is about these TFs is that a lot of them have not been very well characterized. In fact, we only know binding motifs for around half of the TFs in this family. So this is what a canonical C2H2 zinc finger TF looks like. Like you have the protein and inside which you have a number of zinc finger domains. So each of these zinc finger domains to basically recognize DNA, and they recognize around 3 nucleotides of DNA. And each zinc finger transcription factor basically can have a number of these C2H2 domains inside them. And sometimes some of these TFs have as many as like 39 zinc finger domains. So what does this mean? This potentially means that you can recognize really long sequences and really complicated sequences, potentially like using lots of combinations of these zinc finger domains. So this is a typical structure basically of a C2H2 zinc finger domain. You have an alpha helix coordinating with a zinc atom, and there's specific locations on this alpha helix which determine the specificity of the zinc finger domain to a trinucleotide of DNA. And so the question is, like, if we know the amino acid sequence of like a zinc finger domain or a sequence of zinc finger domains, can we roughly predict what kind of DNA sequence it would bind to, right? And this kind of mapping of amino acid sequence of the zinc finger protein to the DNA sequence that is actually bound is known as a recognition code. And the specific mapping is called like the zinc finger recognition code. So there has been a lot of prior work to try to decipher and understand what this zinc finger recognition code is. So what these approaches have done is they've taken this system known as the bacterial one hybrid system, and they've expressed this zinc finger transcription factor called EGR1, and they modify it has 3 zinc finger domains in it. So they just modify the last zinc finger domain to be coming from a natural library of maybe 47,000 naturally occurring zinc finger domains. And by measuring the change in specificity, by changing these zinc finger domains, you can learn the marginal binding of each of these naturally occurring zinc finger domains. Okay, so maybe if you have like a new zinc finger transcription factor now, like ZNF10, and you know what domains are there inside them, you can concatenate all these predictions, and you can have like a recognition code predicted motif. So the question is like, okay, so how well does this approach actually work, like for a real protein? Because like, this approach does not take any interactions into account. And of course, it does not take the full protein and all the cellular context into account. So like, does this approach work, right? So we'll come to that. But just before that, the thing to note is that what it predicts is like, if you have a protein with n zinc finger domains, it would predict like a motif with 3 times the number of domains, because each domain on average is supposed to be binding 3 nucleotides of DNA. So, okay, so to actually study the binding of the full protein, you need to do either in vitro experiments like HT-selects, or if you want to understand how they bind like in vivo, you have to do like experiments like ChIP-seq or ChIP-exo. So there have been pretty large scale studies done on some of these zinc finger TFs. So there's like HT-selects from the Tapale lab on like 120 C2H2 zinc finger transcription factors. And ChIP-seq and ChIP-exo typically also is pretty hard to do for these transcription factors. The reason is it's hard to find antibodies that target these natively. So how people have gotten around that is basically by creating these constructs, which are fusions of the zinc finger TF with EGFP. Then you express them in HEC293 cells, and then you can target the chip basically. So you can target the EGFP. And so there have been studies from the Hughes lab and the Trono labs, which have done this on hundreds of C2H2 zinc finger TFs. Now, as part of ENCODE, and it's a collaboration between the Snyder and the Hughes labs, we also did ChIP-seq basically on 208 more of these C2H2 zinc finger TFs, of which 106 are new. And all of this is now available on the ENCODE portal, and it's all processed. And but then all in all, like we have in vivo data, like using all these data sets, previous studies and our new data set on like 422 C2H2 zinc finger transcription factors. So this is ChIP-seq and ChIP-exo. So, okay, so once you have this in vivo data, let's say you want to learn motifs from this in vivo data and try to understand what kind of sequences to bind to. So you do motif discovery. Now, it turns out that like traditional motif discovery methods are like MEME don't work very well for these C2H2 zinc finger TFs. The reason is that these TFs, they bind these, they bind a large number of repeating families, which makes like motif discovery difficult. So what has been found to work well in practice is this method called recognition code assisted motif discovery, which basically biases the motif discovery to be like looking for sequences close to the recognition code, and that assists it in actually finding better matches. And this works pretty well. So, and this is from the Hughes lab. Okay, so let's say we look at all the motifs that are out there for the C2H2 zinc finger TFs. So be it like HT selects derived motifs or maybe like recognition code derived assisted motifs. So let's look at them. And so we look at this database called cis-PP, which has like a ton of these motifs, right? And what I've done is I've plotted the length of these motifs versus the number of zinc finger domains per transcription factor. And remember, like what the important learning from the recognition code was that if you have like n zinc finger domains in your TF, you should be having something like 3 times the number of zinc finger domains per motif if it's using all of them. But what we see is that the length of the motif does not really seem to depend on the length of the on the number of zinc finger domains. In fact, like it's quite flat. There's no linear correlation. So like what's going on? Like when you have a lot of TFs with like so many 20, 25 zinc finger domains, like why are the motifs so much shorter on average? Like what are these extra zinc finger domains doing? Like are they even binding DNA? So we try to understand some of these things, right? So to try to have some kind of understanding, you can look at the best studied C2H2 zinc finger TF, which is CTCF. So what is known about CTCF is that CTCF is known to bind this canonical motif, and this is the core motif of CTCF, and this is known to be bound by like mutating different zinc finger domains and doing experiments on them. So this core is known to be bound by zinc fingers 4 to 7 of CTCF. But however, like there have been studies which have found this basically this upstream motif that's present to the core, but this is only present on like 10% of the binding sites. And this upstream motif has been shown to be bound by zinc fingers 9 to 11. So the question is this, like, is this sort of phenomenon like more prevalent? And is this happening with other zinc finger TFs as well? So maybe there could be like, these other rarer motifs which are bound by other subsets of the zinc fingers, but maybe we've just been missing them out because maybe our motif discovery tools are like not capturing these kinds of motifs. And so this also raises the other possibility, like if you're binding with, apart from the core set of zinc finger, if you're binding with like other subsets of zinc finger domains, like maybe you could have like multiple binding modes using like different configurations of zinc finger domains. So the question is, how often do these kinds of things happen? Like, so why are the motifs shorter in general than what predicted from the recognition code? And like, do we have like situations where we have like multiple binding modes using an alternate motifs, right? So to answer these questions, basically we trained BPNet models like what Anshul described on all of this ChIP-seq and ChIP-exo data. And once you train the BPNet models, you can learn you can distill motifs out of them using this TF-Modisco algorithm, which uses the importance scores from the model. And we learned two kinds of motifs. So one of them is, of course, the position weight matrix, which summarizes like frequencies at individual nucleotides. And the other thing is the contribution weight matrix, which is an aggregate of the contribution scores, the predictive contribution scores at each base. And this distinction will be is quite important and would be more important at the end of my talk. So, okay. So just a second. Yeah. So this basically summarizes. So we trained a model for every TF and every data set. And this basically shows you like the performance, the aggregate performance of the model on the holdout set. So what I'm reporting is the Pearson correlation between the observed and the predicted tracks. And as you can see, it's like pretty generally high on the holdout chromosomes, like across the board for every TF on every data set. So once you have the models, you can learn the motifs. And I'm going to show you like we recover known stuff. So here I've compared it to some of the HT selects motifs, and you can see that the HT selects motif is shown on top. And in general, we can recover like the HT select motif pretty well from the in vivo data, like using this TF-Modisco algorithm. We then compare it to the other gold standard for zinc finger motif discovery, which was the recognition code derived motifs. And we see that we can generally recover the recognition code derived motif. And I should point out that we're doing it without assistance from the recognition code. We're doing this de novo. So, okay, so we can recover known stuff, but so how do we learn something more, right? And how do we quantify that? So we realized that it's actually quite useful to take your to take the motif that you learn, the TF-Modisco motif, and align that to the recognition code So what I mean by that is you take your motif that you have learned, your in vivo derived motif, and then you do a Smith-Waterman dynamic programming alignment, which allows for gaps, and you align it to this B1H recognition derived motif. Okay, and that's pretty useful because if you have this alignment working, right, what that tells you, like if you see this alignment, it would tell you that, okay, this right bit of the recognition code aligns pretty well to the motif. That what that means is that the zinc finger domains that recognize this sequence on the right, they're active, and those are the ones that are binding DNA. And the zinc finger domains that potentially recognize this sequence on the left are not binding DNA in this situation. And also, if you have gaps that are needed to make the Smith-Waterman alignment work, they would give you information on whether the zinc finger domains are like in a more compressed configuration or an extended configuration. Like, so they give you so this gap alignment gives you a lot of information, and we're going to show you that, like what that means. So okay, so we did this gap alignment for every TF motif that we learned for each of these TFs. And we actually see that we learn more than one motif for a large number of these TFs. We learn like multiple motifs for many of these TFs. And so what does that look like? So this is one of the examples. So this is ZNF382. And for this one, we found like 5 different motifs. So I'll go over all of them. So this is the first motif for ZNF382. So for each of them on top, the top row is the B1H, which is the recognition code predicted motif. And the second row is the de novo motif that we learn. So this is the first motif where we've learned a very long motif. And this, from the alignment, you can see that it seems to be using every zinc finger domain to bind this long motif. Then we found a second motif, which only aligns to this central bit. So it's using some subset of zinc finger domains, right? And this is there on like 1/3 of the number of sites as this version. Then we find a third one, which is using even fewer zinc finger domains. There's this GAG that is prominent. It's missing the CAG here, so it's using a smaller subset. And we find a fourth one, which is using the zinc finger domains that align to the right bit of the motif. And we find a fifth one that just uses the central bit. So ZNF382, so it seems like it can bind DNA in a lot of different ways, depending on what kind of sequence is available. And it uses different subsets of its zinc finger domains to recognize the DNA. And among all these motifs, like this main one was known before, and like none of these were reported before, but we're able to identify many more of these alternative configurations. So if you do this for every zinc finger TF in general, and like if you try to map the number of zinc finger domains that are used to bind DNA per TF, if you do this for all the motifs in the literature for cis-BP, like as I reported before, like we find like the number of motifs used to bind DNA per TF is like much lesser in general than the number of domains present. And so you'd see that, yeah, it's like much under the line X equal to Y. But if you do this with our set of motifs, you see that we find many more zinc finger domains are used to find DNA, used to bind DNA. And there's now a linear correlation with the number of domains. So this is for 2 reasons. Firstly, we find like more alternative motifs, which like use different configuration, like which use different subsets of zinc finger domains, and these are missed out previously. And secondly, we generally just find much longer motifs, which were like motifs previously were getting cut off arbitrarily. And when we do this alignment to recognition code, we can actually identify like more number of zinc finger domains used to bind DNA. And if you like directly compare the number of domains predicted to bind DNA before with our methods to as predicted before, you see that in general, we predict many more domains binding DNA per TF than previously shown. So now I'm going to get into a few case studies to showcase some of these things like in a lot of detail. So the first case study I'm going to go into is ZNF140 and its alternate binding modes. So ZNF140 is this zinc finger TF with 10 zinc finger domains in it. And the top row is the recognition code motif. The second row is the TF Modisco learned motif. And we call this the compressed motif. And we call it compressed because, as you can see, it's slightly compressed with respect to the B1H motif. There's a 6 base pair gaps in the recognition code, and there's a 5 base pair gap between the G and the A here. And this motif is like what is actually also known in the literature for ZNF140, and it's the more frequent one. But then we also found what we call the regular motif. And this is much rarer, and it's a new motif which has not been reported before. And this is only again found on 10% of the sites. And as you can see, it's not compressed. It's the same length as the recognition code motif. So we wanted to quantify this better. Like, can we say like what's going on energetics wise? So for that, we did this assay known as SpecSeq. And so this is an in vitro assay which can basically measure energetic affinities with like very high resolution, with like 0.1 kT binding resolution. So we did this with the help of the Polyfordis lab at Stanford, and her postdoc Zhang helped us to do this experiment. So essentially what this does is you design a library of DNA sequences you want to test, and you put this in a gel with your protein of interest, and then you apply an electric field, right? And the stuff that is bound and the stuff that is unbound separates, and you can sequence this what is bound, and then you can basically learn uh, affinities, right? And those would give you like energetic affinities after that, based on like what is bound or not. So so in the bound sequences, we're able to find both motifs. We're able to find the compressed motif. We're also able to find the regular motif. So now that you have like energy representations of these motifs, you can use them to score every member of the library, right? So okay, so let's say we take the compressed motif and we score every library member, right? So you see that generally it's a straight line, but there's this blue section here which is like under the line y equal to x. So this is actually not energetically explained by the compressed motif. So it has additional stability. So this turns out to be sequences that come from the regular motif. And if you use the regular length motif and you score sequences, you again see you can't explain the extra stability of the compressed motif, the compressed sequences. But if you take the minimum energy from both models, you get this perfect straight line. So what this means is that either.
Sorry to interrupt you. You have about 1 minute left.
Oh, okay. So okay. So I think I made the point here, so I'll skip ahead. So okay. So let's go to ZNF. Let's go to ZNF10, right? So in ZNF10, we found like there's this model with like zinc finger skipping, because we see that you need to do this gapped alignment, you need to actually skip a zinc finger. So we actually validated this with SpecSeq as well. And we tested a lot of like spacing preferences, like 1, 2 base pairs in our library. And what we found is that actually the compressed motif, which actually skips an entire zinc finger, which we found with our model, is the most stable. And like having these spacings like actually reduces the energetic affinity. And our final case study was ZNF121, which binds like transposable element sequences. And I'll probably skip that in the interest of time, because I don't think I have enough time to go over this right now. But in generally, like we did like affinity, we did like two, like maybe I can just show you this. Like we find like we have this contribution weight matrix for this motif, which has been shown here, which is the CWM derived motif, and I've aligned it to the recognition code. We also have the PFM, and the PFM is pretty different. And what we see is that there's a lot of flanks, which the CWM says is not very important for binding. But when you look at the affinity-seq derived motif, which is like an in vitro assay which uses genomic sequences to learn affinity, it recovers the PFM. We then did HT-selects, and HT-select starts out with a random library, right? So it's a truer representation of the energetics. And the HT-selects actually matches the neural net learned motif really well. So this is pretty interesting, because what this shows you is that the neural net can denoise like repeating frequencies in the genome, and it learns true binding affinities. And it's a better representation of binding than just raw frequencies. And so, yeah, so it's very powerful, this modeling approach, and because of this we have been able to really dissect binding of these zinc finger TFs, those that even bind like these transposable element repeat families, and this was not able to be done before for these zinc finger TFs. So yeah, so I've come to the end of my talk. So basically we've trained like BPNet models, we've learned a lot of new, characterized a lot of new C2H2 zinc finger motifs, and we found like multiple binding modes, skipping of zinc fingers. So this expands the human TF lexicon. And thank you. I'd like to thank all my collaborators and my funding sources. And yeah, so maybe I can take some questions.
Brilliant talk, Abhimanyu. Thank you very much. Sorry to put you under some time pressure there. So because we only have 17 minutes left in the session, I'm going to ask you to answer your questions by typing in the Q&A, and then we'll move straight on to Daniel. And if Daniel can get his talk done in less than 17 minutes, I know that's a bit of a big ask, we'll return to some of the questions. But if not, please do type your answers. Thank you very much. Daniel, take it away, please.
You're muted, Daniel.
we go. Sorry about that, guys.
No problem. We can hear you now.
Okay, great. Okay, thank you so much for having me. With the limited time, I do want to be respectful of it, so I will be speeding through a little bit. But I think Anshul and Abhi gave great talks that have set me up really well. So I think you're all prepped conceptually for the deep learning work that we've been doing. And I'll be talking about that applied to the skin differentiation system. So the epidermis is the outermost layer of your skin, covers your whole body. It's a linear terminal differentiation. So you have the basal layer of skin stem cells at the bottom of your skin that then migrate upward and differentiate to become eventually the cornified layer of skin. And we have a great model system of being able to take those primary cells, grow them in a dish, induce with calcium to basically have them differentiate into that terminally differentiated keratinocyte. That happens over a course of 6 days. And so for that, we were able to collect a variety of different genomic assays. So these are bulk assays. ATAC-seq, PAS-seq is 3' RNA sequencing, 3 histone mark modifications, K27 acetylation, K4 monomethylation, K27 trimethylation, as well as high-chip for contact and looping information as well. We were able to have the RNA-seq and the ATAC-seq at high resolution about every half day across that 6-day time course. And then we did the histone marks and the high chip at day 0, day 3, and day 6, corresponding to the stem cell state, the progenitor state of keratinocytes, early differentiation, and late differentiation. So what this was able to provide for us was a really rich and dynamic view into differentiation in the epigenetic space as well as the transcriptomic space. So what I'm showing you here is a locus around integrin beta 4, which is a component of hemidesmosome assembly in early differentiation. And you'll see that this gene decreases in expression across time, and a nearby region linked by high chip is showing decreased accessibility across time. And there are a variety of other loci that have interesting and dynamic epigenomic windows around these genes. So with all this data, we were able to then collect it and identify which regions were dynamic across time. So we found about 40,000 accessible peaks that were dynamic in 15 different dynamic patterns across time. We were able to look at the acetylation and the monomethylation around those regions and able to see what those patterns of histone marks around these regions were, as well as to look at functional enrichment within these regions. So we were able to dissect the epigenome, figure out which parts were dynamic, what the histone marks looked like, and a little bit what the functional purposes of these regions may have been within skin differentiation. We also had the RNA-seq data, the PAS-seq data, and similarly, we were able to look at the transcriptome and figure out which genes were dynamic and what those different patterns were, and subset them into 11 different groups of genes, as well as the different functional enrichments for those different modules of genes. So we have this large compendium of integrative genomic data. And as you know, we're very interested in what's happening within the DNA sequence. So within a window of the genome that has accessibility on it, we know that underneath that there is regulatory DNA. Within that there are motif elements that are recruiting transcription factors that then will modulate downstream gene expression. So how do we look at that DNA and figure out what's happening within the DNA? We apply neural nets to it, as well as talked about by Anshul and Abhi. So the input is DNA sequence into a convolutional neural network. This is a multitask output where each task is going to be a different time point of data. So basically we have like a day 0 time point task where that predicts the level of accessibility within that day 0. We have a day 6 output task, which predicts the output of accessibility at that time point. And so we have a multitask model, DNA input predicting the molecular phenotype, the accessibility on top of it. So how do we actually train something like that in a robust and accurate way for a convolutional neural network? We actually start with the ENCODE and roadmap data. So with the large compendium of data that we have, we can ask on any genomic window, what are its predictions for DNase or ChIP-seq within the ENCODE roadmap compendium. And this comprises about 400 or so DNA-seq experiments, as well as another 1,500 ChIP-seq experiments. And that helps the model to kind of get a sense and understanding of what is regulatory DNA. From there, we trained the classification model. So can the model now more focus more specifically on keratinocyte information and predict for a genomic window, whether that window is accessible or not, so a binary classification. And then from there, we asked it to do a regression task. So can we actually have it predict the level of accessibility, not just whether it's accessible or not accessible. And so when we do this training procedure, it actually works pretty well. What we find is actually that when we do this transfer learning procedure, we have much improved performance, as well as much more robustness in that across 10 folds of data. So that really gives us a lot of confidence that this is working effectively. And just to show you what that sort of correlation actually looks like, here are outputs from 3 of the tasks, day 0, day 3, and day 6. And you can see what our predicted looks like compared to the actual. And also just to show that this actually works really well across time as well. So when we look at our predicted outputs from the neural net for the dynamic ataxic peaks, we actually do really well in comparison to what was observed in the ataxic signal as well. So this gave us a lot of confidence that the model that we had trained on the epigenetic data within skin differentiation was doing well, was accurate and robust, that we could then interpret. So for interpretation, we've talked with Anshul and Abhi about backpropagation methods, deep lift, where we go from the output task backwards through the model and basically figure out what the base pair contribution scores were on the DNA sequence. So for a specific sequence and a specific day, given that our output tasks are different days of time within our system, what was the contribution of each base pair to that final output prediction within accessibility? So as an example here is a genomic element linked to the promoter of keratin 77. This is a gene that is increased in expression across time. And we can do that backpropagation for each of the different output accessibility tasks. And what this gives us is multiple signal tracks with per base pair scores. So you can see that certain base pairs have basically been downweighted and zeroed out, where others have been increased in terms of, or upweighted in terms of their importance to that task. So starting from day 0 in our time course all the way through to the end of differentiation at day 6. And what we nicely find is that these scores look a lot like motifs. And you can also see dynamic motifs as well. So P63, for example, a known key regulator within skin differentiation, shows up as equally important across time, whereas CBPA, which actually does increase in expression across skin differentiation, has a dynamic motif here that is increasing in its importance across the differentiation process. So we're able to get these kind of dynamic outputs in terms of per base pair contribution scores, also known as importance scores. And we wanted to know whether these meant anything functionally. And fortunately, within our system, we do have ChIP-seq as well as our deep ATAC-seq from this, this compendium. When you look at the ChIP-seq data, if you subdivide basically by the motifs that were kept by the neural net, were outweighted by the neural net, versus the motifs that actually were zeroed out by the neural net. So these are matched by the actual PWM score, as well as by the GC content around that PWM. What you find is there is a lot more ChIP-seq signal over the neural net active motifs compared to the neural net inactive motifs. So there's more occupancy based on the ChIP-seq for the neural net active versions of the motifs compared to the inactive versions. And I'm showing 2 examples here with P63 ChIP-seq, as well as ZNF750 ChIP-seq, where we see that the neural net does seem to highlight motifs that are actually occupied by TFs. Now, our ATAC-seq was also fairly deep, so we could do something similar there, where we asked if you subdivide by the neural net active motifs, again, the ones highlighted by the neural net, versus those that are inactive according to the neural net, what you see are 2 interesting things here. One is that the accessibility flanking the footprint is increased around the sites where the motif is active, as you would expect if a TF were sitting there, as well as the footprint is deeper as well too. And so both the ChIP-seq as well as the ATAC-seq footprinting gave us confidence that what the neural net was highlighting was something that could actually be functional and could be meaningful in functional genomics. So looking now in particular at these neural net active motifs, What we were then able to do was try to pull together a compendium of which motifs were actually being used within the dynamic epigenome, as well as then which transition factors were being used. And just for the sake of time, I'll kind of go through this really quickly, but essentially for each trajectory, we can look for we can do these backpropagation methods to get the per base pair contribution scores, do motif scanning, and then get a landscape of which motifs were used in the dynamic epigenome, as well as when they're important in the epigenome. We can correlate that to transition factor gene expression to then get a landscape of which transition factors we also believe were important. Okay, so we can go a little step further than that, because with these models now, we can then do, as Anshul did, we can mutate motifs within sequences, as well as use synthetic sequences to then figure out whether any of these motifs are working together. So if you take a genomic instance, you can do these combinatorial mutations. You have your original sequence with motif red, motif blue. You can scramble one, scramble the other, or scramble both. And then you can run those through a neural net. And then you can ask, okay, compared to having both motifs in there and that predicted joint effect by the neural net, if you had only the motif red or only the motif blue, and you look at the log additivity of that, how does that actually compare to that joint effect, right? So log additivity being multiplicative in the original space. Is this joint effect going to be super multiplicative? Is it going to be multiplicative? Is it going to be submultiplicative? And these are questions that we can ask because of a good, strong model that can make these predictions based on these adjustments to the DNA sequence. We can also do the same thing with synthetic sequences. And so we looked at both genomic examples as well as synthetic versions to compare those two. Here's a vignette that basically tries to illustrate what we're doing for a specific example. So here's a gene and a region linked to it. And what you can see here is here's the original per base pair contribution track for that DNA element. You can take out the GRHL motif, you can take out the CVPA motif, or take out both. And then you can basically get scores for each of these different new sequences, right? So here's a scramble. Here's your baseline accessibility predicted by the neural net model. If you just have one of the motifs, here's the baseline, here's the predicted accessibility. If you have just the other one, and then you have it both, and what you're seeing here is probably something that looks a lot like a multiplicative or a log additive effect predicted by the neural net model. So we can do this across a variety of sequences for a variety of motifs. Essentially, what we find here is that when we do that, we actually can reduce down from the co-occurrence matrix of motif hits that co-occur down to a more specific set of pairwise interactions. Below the diagonal, I'm showing you the genomic data, pairwise interactions based on the genomic instances that we had mutated. And above the diagonal, I'm showing you the synthetic data where we had taken background sequence and inserted the motifs, showing you how those two results compare. One quick note I'll make about this before we move on is that when you just look at co-occurrence of motif pairs, if you look in accessible regions, there's just a ubiquity of motifs. And so it can be quite hard to figure out which ones are actually working together versus which ones are maybe just present. And we found that our method really helps narrow down for us which ones might actually be functional pairwise interactions. So this is all still an epigenomic space since we did all the predictions and everything within that space, but we were wondering whether we could actually learn whether these were actually doing things in expression. So we characterized these things using an MPRA experiment. We took these pairs of functional rules that we had gathered from our epigenomic analysis. We took those genomic instances, we did the mutations and threw them into an MPRA experiment into the cells and then harvested at days 0, 3, and 6 across the skin differentiation model system to figure out how those elements changed across time. So with that sort of a setup, what we could test were two key things. The first thing was, do those elements, based on how we had collected them and gathered them as examples of pairwise motif combinatorial rules, do they have the expression that we expect across time? So here's an example where we looked at fragments that have the Hoxsey motif and the ERG motif, about 19 examples of those. And then we also looked at those compared to their double scramble, where we had mutated both examples of those motifs. And what you can see here is that for those endogenous examples, you see a consistent decrease in expression based on the reporter activity across time within our differentiation process as we expect for these elements. And when you do the double scrabble, when you remove the Hoxha motif and the erg motif, you actually are able to ablate that signal, and you lose that dynamic pattern that you have for those endogenous elements. We're also able to look at the combinatorial activity. So if you just have the erg motif, what does that expression look like? If you just have the Hox expression, what does it look like? Versus the Hox and erg, what does that expression look like? And for the sake of time, I'll skip through these here, but these are just some other examples that show similar ideas, but different trajectories across time. And to summarize, basically out of 80 different pairwise interaction rules that we had tested, 55 of those actually panned out with either a multiplicative or a supermultiplicative effect. And so you can see that here in summary. So a good number of these actually worked in expression space, that we had first pulled them out in epigenetic space. And that leads to this validated combinatorial motif lexicon, where these are the 55 rules here. What this is showing here is which motifs are involved in that rule. This is the accessibility on top of those instances where that motif pair is present within the skin epigenome. This is the downstream target gene module that is controlled and the expression of the target genes downstream. The MPRA data is shown here, what the interaction is, whether it's supermultiplicative or multiplicative, the predicted versus the actual, and the MPRA results across time, and then functional enrichments as well to show that these rules do carry out different functions within skin differentiation and give us a little bit higher granular resolution into what these rules and what the circuitry may be doing. And then we, since we have these rules and we have connections to predicted TFs, we were able to look at the network to say what is our predicted functional network of TFs or the circuits of the networks that are involved within this, from progenitor cell maintenance through to early differentiation through to late differentiation. I'll skip this for time. And basically just to summarize, we basically took this as a way to demonstrate a generalizable framework for dissecting cis-regulatory logic. So if you look at we just a taxic and RNA-seq, we can basically build a model on top of that with epigenetic data. We can then infer combinatorial rules by getting the importance scores, getting a motif set, getting combinatorial rules, running those through an MPRA validation to then figure out what those rules are actually doing in expression space to control downstream gene expression. So you can go from TFs to what's happening in the DNA to what's happening downstream in terms of function. I'll save the summary since we're at the end of time. This is a bioRxiv preprint up online as well too, so feel free to look at that if you'd like further details. And also and finally, really quickly, I'd like to acknowledge that this really takes a village to carry out a project like this. So I'm very grateful to all the labs that were involved in the data generation and the computational and all the comments and help. And that's all. Thank you so much.
Daniel, we are completely out of time. I'm so sorry. I see you've got your contact details up, so I assume that means people can contact you.
And people, if there are questions, I think these guys could stay on for a couple more minutes to answer any more questions to make sure they get them all. But that was a real tour de force. And
yeah, I should apologize. I hogged way too much time, so I made it difficult for them. That's
my intro was too long.
I also want to quickly make a plug just for Daniel. He's also graduating this year. He's an MD PhD student. He's definitely on the postdoc market as well. So if people are interested, definitely contact both Abhi and
look us up. Thanks so much, Anshul. Thanks so much, folks.
Thank you.